Let's talk a little bit more about hair. We've already described how it's, useful, especially if there's the little skin tag that gets pulled out. We'll talk about that. There can be some DNA in there, so that can be useful. If not, we're sort of relying on morphological evaluation, and we can't really ever reach that individualization using that. Just a little bit more on hair and its morphology. Hair grows out of the root and follicle, comes through the skin, and there's a lot of variety here, and we'll talk a little bit more about the anatomy of the hair shaft itself. So the hair shaft itself comprises of the cuticle, and that's the outside covering. It's made out of these overlapping scales. The cortex, which is the inner layer, made with keratin, and also the pigment is involved inside of the cortex. The medulla is this center layer running down the center of the shaft itself. Now the cuticle is kind of great because it's covering. There's scales that cover this structure, and you can get these different scaling patterns. And the scaling patterns can be sometimes unique to the species that, so you can actually, try to identify human versus animal in this way. And so here's some high-res images, probably with a scanning electron microscope, where you can see this is what cattle, otter, or human scaling pattern looks like. You can tell they're pretty different. So that can be useful, and just a way to rule out or what type of hair it is. The cortex, which is the main body of the hair shaft, again, it has the melanin, which is little pigment granules, and that's what gives hair its color. The medulla, again, is, like I mentioned, is the center column running through the center of the hair. There's different types of this banding pattern where you can have no banding present, interrupted, continuous, or fragmented. And so those can just be the more sort of characteristics that you can identify in the hair. There's actually an index, so the medullary index, which is the diameter of the medulla relative to the diameter of the hair shaft. And in general, for humans, you're looking less than 1/3 of the shaft diameter, and for animals, the medullary index is generally greater than 1/2 the shaft diameter. So you can kind of see that these ratios of the medulla relative to the whole shaft of the hair itself can also be diagnostic. It's also important to note that hair grows in 3 phases: anagen, catagen, and telogen. So the anagen, and then the catagen, and then the telogen phase are all specific phases of hair where you have growth and followed by the hair actually falling out. And so that type of evidence can be significant as well, because if hair is yanked out forcefully, that could be information that would probably pull out more biological material versus the hair following its natural path and falling out on its own. So that root, that chunk of skin that maybe could get pulled out from the scalp or wherever, this is described as the follicular tag, and so it's a translucent tissue surrounding the hair shaft near the root. Okay, and then there's going to be some nucleated cells that can contain the DNA. So identifying characteristics, we can get at species like I described, the medullary index, cortex, color, the scaling pattern, so all that can be useful. And so, we can look at traits like color, length, and root diameter, medulla structure, condition, and hopefully that can be more information that's useful. So, what kind of other kind of questions that are asked with hair analysis? Well, you know, can a hair indicate, you know, an origin? Can racial origin of hair be determined? Some of these are more difficult than others, but you know, this is kind of like what we're trying to get at with hair analysis. Now, let's talk about fibers for a minute. So, fiber is different. Fiber is the smallest unit of a textile material. It is comprised typically of polymers that are shaped into filaments. So, aside from, well, even cotton would have a fiber, a sort of smallest unit of that material. So just think of all the fabrics and clothes that you have. Those could be blends of different fibers, and or a single fiber, and so that can be useful as evidence. So how it can be used? Well, you know, perhaps you know, if there's other trace evidence, you know, then fibers can be transferred to and from a person or object. So that might be able to link, you know, a person to a crime scene, or at least a person to that area, and so that can be something that's useful. For fibers, they're typically classified into natural and man-made. So man-made fibers are polymers, synthetic and natural, and then natural fibers come from animal, as you can imagine, like wool, vegetable, as you can imagine, cotton, and then mineral. Which, actually, off the top of my head, I can't think of one that would fit into that category. Uh, However, we'll keep moving forward. So, natural, again, found in nature, not greatly altered. Synthetic, manufactured with fiber-forming substances. Semi-synthetic, modified with natural materials. You know, there's you know, interesting ones that we've described already, but others would be cashmere, wool, camel hair, hemp. Okay, acrylic would be another one that I haven't mentioned before. So all these can be fibers. Usually, you can analyze this stuff with a stereo microscope or a compound microscope. So what is the value of fiber evidence? Well, depending on the ability of the criminalist to identify the origin of the fiber or narrow the possibilities to a limited number of sources. So that can be very useful. Microscopic comparisons with, you know, questioned, known, and unknown. There are reference libraries, but you can imagine this would be difficult to reference from a library. Other morphological features that could be important in comparing fibers are length, what their length is, presence of delustering particles, cross-sectional comparisons. And, you know, there can be a lot of diversity here, so that can actually end up helping you. So methods for fiber comparison, compositional differences may exist, and so we can use visible light microspectrophotometers, chromatography, not that much. Infrared spectrophotometry can be really important, and we'll talk later about refractive index as well. These are examples here below of you know, the FTIR analysis. That can actually be really useful. So another thing we can do for you know, different fibers are using the polarized light microscopy. So actually looking at different fibers under different light filters, we can actually get some more information about the composition of the fiber. Since fibers are mass produced, we really can't get at individualization, and there's not much of on the order of statistical databases for this stuff. But you know, associations can be helpful. Here's an example. In Atlanta in the early 80s, 30 boys missing or murdered. Similar fibers were found on all the bodies. And these carpet fibers were unusual, you know, and so perhaps they were, you know, sort of rare. And so, you know, basically, you know, all this whole case revolved around this, you know, blue rayon fibers that were found in this throw rug that were uh, super unique, and that actually cracked this case. So fibers can actually be useful. I have for this one, and I'll have another lecture shortly. Thank you so much.